minutes. Let us wait 2 minutes more.
Okay.
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 Let me see. Yeah, okay. That is, are you a veterinary faculty there?
Yes, sir.
And are these veterinary students, or what area, where are they at in studies?
Yes, sir. 90% will be the veterinary students. Of course, few biology students will also join the talk.
Great.
And some field veterinarians also there, sir.
Wonderful.
So,
Now, also, are your students mostly from local, or are they from all over India?
sir, mostly from this local area, but some are from outside this area also.
Mm-hmm.
Yeah.
Mostly from the West?
Mostly from Northeast, sir. This area coming under Northeast. Yeah.
Or East, I'm sorry. I sometimes get turned around. I was able to visit Kaziranga National Park on my last trip. It's a brilliant, brilliant park. Beautiful.
Basically, sir, sir, can we start?
Yeah. I think so, yeah. It's more than 150 participants, so we can go ahead.
Yeah, it's above 150. I feel we can start only.
Yes.
So, good evening all. I welcome all of you to this international webinar. So today is the first day. Yeah, in order to, I request all the participants to keep the mic mute during the session. And in order to start with the program, I request Professor P.C. Kalda sir to propose welcome speech. Please, sir.
Thank you, Dr. Rajesh. Am I audible?
Yes, sir, you are audible. Yes.
A very good morning to Professor Eramsay, sir, and good evening to all the dignitaries of Central Agricultural University, our Dean Madam Mar, my colleagues, dear students, participants from various parts of the country. On behalf of the College of Veterinary Science and Animal Husbandry, Central Agricultural University, Aizawl, Mizoram, I welcome our speaker, Professor Eramsay sir, who is a renowned professor and former student of University of California, Davis. I am grateful to him for accepting our invitation to deliver a talk of today's event. Tonight, the topic of presentation of international webinar is canine distemper. So canine distemper has been recorded in domestic dogs for centuries. It is now recognized as a worldwide problem of carnivores and has the second highest fatality rate of any infectious disease after rabies in domestic dogs. Canine distemper virus, it invades the nervous system and replicates the neurons and glial cells of the white matters during a period of severe viral-induced immunosuppression. Demyelination occurs in infected white matters area in the absence of inflammation. Tonight's speaker is known for his work in canine distemper. Dr. Eramsay sir traveled abroad on more than a dozen large-scale projects where he helped establish solution to the problem. I had listened to his presentation for several occasions, including the latest one at the Center for Cellular and Molecular Biology, CCMB, Hyderabad during 2019. Accordingly, I was able to make him a request to give a talk on this issue for veterinarians studying at various institutions as well as working at different organizations. I feel extremely honored and excited to introduce the speaker of tonight, Professor Eramsay sir, who is a renowned zoo veterinarian and animal lover, and after all, a teacher. It is such a privilege to have you, sir, on the stage as a speaker. Dr. Eramsay is a native of California, and he received his DVM degree from the University of California, Davis in 1980. After a year in private practice, he became the first resident in zoological medicine at the University of Tennessee, Knox Valley. After leaving Tennessee, he was the staff veterinarian at the Oklahoma City Zoo for 5 years. Dr. Eramsay also worked in the pathology department of the National Zoo, Washington, D.C., and as assistant director of the Sumatran Rhinoceros Trust, a capture project in Indonesia. From 1988 to 1991, he taught clinical exotic animal medicine at the University of California, Davis. In 1991, he became board certified in zoological medicine and returned to the University of Tennessee College of Veterinary Medicine. From 2007 to 2008, Dr. Eramsay sir was the president of American Association of Zoo Veterinarians. He is the author of numerous publications related to zoo medicines. Dr. Eramsay retired as a professor in the Department of Small Animal Clinical Sciences in 2020. His research focuses are captive and free-living wildlife and non-domestic feline medicines. Now I like to invite Professor Eramsay sir to deliver his talk. Professor Eramsay sir, please.
Thank you very much for the introduction, and thank you all for coming in. I'm going to attempt to share my screen now. Let's see how this works. Good. Does that show up okay? And can people hear me all right?
Yes, sir. You are clear and it is audible.
Wonderful. Well, thank you very much for the invitation. It's nice to be able to visit India, if only virtually. Again, I look forward to when I can return to your country in person. I was asked to speak, and I decided to talk a little bit about canine distemper because I think it's an area that I've spent quite a bit of time looking at, and it's an area that I think is it's a disease that is becoming more and more important in a variety of species, and so we'll go from there. As an overview of what I'd like to talk about, I'm going to give you just a little bit about my background in addition to the introduction. I'm going to talk a little bit about canine distemper virus, the microbiology. I'm a clinician. I'm a person that kind of sticks needles in animals and treats things. I'm not a microbiologist, so that's going to be brief. I'm going to talk about host range a bit, and then focus mostly on canine distemper and FELIDS, which is the area that I've been working most closely, because they're an excellent example of what happens when this virus goes into a spillover host. And then actually talk a little bit about vaccines, another area of my personal interest. So from a background standpoint, most of you probably do not know where Tennessee is. So this is the United States, and Tennessee is located in what we call the central area of the southeast, and Knoxville is in the easternmost portion of the state. It's a beautiful area. We have mountains and a national park that is a national biosphere site, but probably most people know us because we're the home to Dolly Parton, the singer. For the last 30 years, I've been associated with the University of Tennessee's College of Veterinary Medicine, and the Knoxville Zoo is a client of the veterinary school and has been my principal venue for working for many years. It's been a client of the school really since the school was instituted, and has historically had a really large collection of non-domestic felids. About 20 years ago, we started working with a sanctuary called Tiger Haven. Tiger Haven now holds about 200 and actually they're up to almost 300 exotic felids. The vast majority of those animals are tigers, followed closely by lions, leopards, and mountain lions. They've been a client of the school for almost 20 years now and have represented a fantastic opportunity to learn and study about big cats. And most of the work that we've done research-wise has come out of working with Tiger Haven. So my story begins actually with respect to this disease. In 2013, we hosted an international conference on wildlife diseases, the Wildlife Disease Association. And during that conference, a couple of colleagues came up and reported about canine distemper in tigers in the Russian Far East. And this video, which I hope is playing well for everyone, is from YouTube, and it demonstrates a tiger behaving very untiger-like in Russia Far East. And subsequently, they began to discover that many of these animals that were having abnormal behavior and other issues were having canine distemper. And I went ahead and I went and approached my colleagues, Doctors Makalous and Gilbert, and said, I have a large collection of big cats, so especially tigers. Maybe we can work together. And that's really where we've used our captive population to come up with some ideas for possibly helping the wild populations. So canine distemper, or maybe a better term would be carnivore distemper, actually has a huge host range. We know all about the canids, and virtually all the canids are susceptible and comparably at risk. So all of these guys get disease and typically show the same kind of pathogenesis that you see in domestic dogs. Probably the ones that are kind of next on the list, at least in North America, are we know that canine distemper, try to use my pointer here, hopefully that'll work. We know that it occurs in the domestic ferret, which is a relatively common pet in the United States, not nearly as common as dogs or cats. The other areas that we think of canine distemper are actually the procyonids. Procyonids are a New World family of carnivores. The poster child here, the one that we see most commonly, is the North American raccoon. This is a kinkajou, this is a coatimundi. And these guys, we see epizootics. In fact, this is the species that I've seen canine distemper most frequently, because we have epizootics of canine distemper in North American raccoons almost every 3 to 5 years. And we'll talk a little bit about why that's of concern. Another species, one that occurs in parts of India, is the red panda, and it is also very susceptible. We'll discuss that, as is the giant panda, which is of very high conservation concern. Probably some of the other animals that are maybe not so well recognized include bears, although cases are very sporadic. Hyenas, again, not commonly documented, but it has been documented. These guys represent the mustelids, which are a group of carnivores related to dogs, and seem to be comparably at risk, although North American river otters here tend to be less vulnerable than some of the others. And then big cats, and big and small cats, including the cheetah, have had documented cases. And then I think the ones that are probably most people don't recognize is there's a whole host of species that have had serologic evidence and occasional cases of disease, but aren't well recognized. So we've seen serologic evidence in deer, in other marine mammals. This is a javelina or peccary. We've seen that outbreak in the United States. Elephants are seropositive. There have been outbreaks in macaques, which is interesting for you folks in India, and even in New World monkeys. So the range is tremendous. So it's been documented in 110 species in over 43 countries now. And so the term canine distemper probably should be almost replaced with the concept of carnivore distemper. And looking at this bottom row, we may even broaden it out more than that. So the virus is a morbillivirus in the family Paramyxidae. It is other morbilliviruses include measles, rinderpest, which is hopefully recently been eliminated. There is a porcine distemper that is closely related, so those are all belong to that morbillivirus group. They have similar signs. They're all very immune suppressive. They're all highly contagious, and they are epithelial, lympho, and neurotropic. They are all neurotropic viruses. So they have similar pathogeneses. The virus is a enveloped single-stranded RNA virus. It is defined by its proteins. So we have a hemagglutinin protein on the surface, a fusion protein on the surface, and those are the two that are most commonly used to identify strains. There is also a phosphoprotein within the virus itself. And there are about 14 strains recognized. And I say about because I fully anticipate that there will be many more identified as molecular biology progresses. This is a busy slide, and I don't expect you to recognize all of it, but this represents one sort of model of how the strains work out. So the strains up in the top, if this was a clock at the 11 o'clock area, the Ondersteport strain and the Snyder Hill strain, these are the original vaccine strains that have been used worldwide. Snyder Hill is no longer used because of its pathogenesis. Virtually all the vaccines that I'm aware of currently use the Ondersteport strain. There are a number of strains in America 1, 2, there's the Asian strain. These represent some ones that we're pulling out of wildlife in Tennessee and the Southeast. Where these strains actually become important is when we start looking at epidemiology and trying to see, so if an animal, a tiger or another species succumbs to distemper, then what strain affected it? And that allows us to look for the reservoir host. To date, all of the strains are susceptible to all the vaccines. So from a prevention standpoint, vaccination should prevent disease from all of these strains. Again, that's a based on not hard science, but on anecdotal information. So what happens when canine distemper enters the body? It usually comes into the body via aerosol, although we are seeing that urine is a highly contaminated biological sample, and so animals may be aspirating or sampling urine. It comes into the body, goes to either tonsils or bronchial lymph nodes, and then multiplies in the lymphoid system. Once it multiplies in the lymphoid system, it becomes viremic and circulates in mononuclear cells in the blood. And then it has kind of two different paths that it might take. So if it goes down one path, it may become, if the animal has adequate host immunity, and that immunity may be due to natural infection or vaccination, the animal may go ahead and resolve the infection without illness at all. A certain number of animals are going to develop CNS signs, and those animals will, or the virus may enter the CNS, and a number of those, very small number, will have CNS signs down the road in older life. This one is difficult to predict because the temporal association between infection and clinical signs is so vague. On the other hand, if an animal does not have a good immune response or is highly susceptible or naive, you have widespread invasion of epithelial tissues in the central nervous system, and that can lead to a severe multi-system disease. I'll show you a few. And in most cases, that's going to lead to death once those animals are showing severe clinical signs. A small number, and when I went to school, that number was around 10%, and I think it will vary with the strain in the region and the populations. A small number will recover, but even those animals will ultimately develop central nervous system signs later in life and succumb. A number of animals may have only mild or inapparent disease, but again, many of those, even though they've cleared the virus from their body, they may still develop central nervous system signs later in life. So in clinical practice, canine distemper is this dog is kind of what I think of as a classic canine distemper case. When I was in practice many years ago, these animals would come in with a poor vaccination history, this kind of greenish ocular nasal discharge. And unfortunately, most of these guys were diagnosed, had to be diagnosed based on clinical signs. Respiratory signs, especially secondary pneumonia, is common in most species, but here we're talking about dogs. And they can also affect the GI system, and you can have vomiting and diarrhea. But certainly that greenish ocular nasal discharge is kind of the calling card of canine carnivore distemper in dogs. If animals have have chronic disease, they may show hyperkeratosis here of the nose and here of the foot pads. And we actually see this, I've seen this most commonly in domestic ferrets. So domestic ferrets come in and seem to harbor the disease for a week or two, and hyperkeratosis, the foot pad, is probably the second most common sign that I've seen in domestic ferrets. In puppies, you may get enamel hypoplasia, as demonstrated in this picture, where the teeth are poorly covered with enamel. They can also have physeal lesions. That's a little less common. Usually an animal, you don't see that because you're not radiographing these guys or the bones on these guys. And then this rather disturbing video is of a dog with what we used to call chewing gum fits or chorea. that is the central nervous signs. Once they start to develop central nervous signs, this disease is invariably fatal. The diagnosis, as I said, diagnosis is largely based on clinical signs and vaccination history. That is to say, lack of vaccination. And in private practice, where money was short and labs were far apart, it was difficult because most of these animals had to be diagnosed just based on physical exam signs and the lack of vaccination without any confirming laboratory tests. Today, at least in our teaching hospital, conjunctival scrapings were the non-invasive method for diagnosing in many of our animals, but actually today in our lab, urine, PCR of the urine seems to be the most common and valuable or most accurate method for diagnosing canine distemper. And this can be done quickly in labs that are set up for it. And certainly in individuals that have died, histopathology and then subsequent immunohistochemistry could be important. So this is my only histopathology slide. It seems obligatory to include at least one photomicroscope slide in every presentation. So this is the brain of an animal, of a dog that has succumbed to canine distemper, and you have a necrotic neuron. And then I think the point to make here is that you can have both intracytoplasmic and intranuclear inclusion bodies with this, is with paramyxoviruses. So this would be the big distinction between the pathology on canine distemper and the pathology on rabies. Going back to the host range, you talked a little bit about dogs. It's something that we're kind of well recognized. In wildlife, probably the animal that has received the most attention is the African painted dog, also called African wild dog. Many people feel this is one of the most endangered canid species in the world. And certainly there are numerous studies that show that the populations have succumbed to a variety of diseases, rabies and canine distemper being the two at the top of the list. I mentioned domestic ferrets before, and procyonids and red pandas. I think the one that is for folks in India, and I don't know about the far eastern area there, but the palm civet and certainly vivarids are susceptible to canine distemper. Another one that is probably less commonly known, this is a picture of a Lake Baikal seal. Lake Baikal is an inland lake in Russia, Siberia, that has its own species of seal. And a population of these guys, which are endangered, succumb to actual canine distemper, which is distinct from bovine distemper. The bovine distemper outbreaks have occurred in the North Sea gray seals population, but the Lake Baikal seals actually had canine distemper. As I mentioned, the other species, mustelids, are well recognized. Bears, almost all the bear cases have occurred in unusual situations, and by that I mean they've occurred in captivity, where the animals have been raised in nurseries with canids or in close proximity to very unusual, susceptible animals in unusual situations. And then cats is what we're going to kind of focus on now. So, and the domestic cat, looking at canine distemper in the domestic cat is kind of interesting, because the domestic cats have lived for thousands of years with domestic dogs, and they seem to have adapted to, in my opinion, the dog disease. So basically, domestic cats do not show clinical signs of canine distemper, but that doesn't mean they don't get infected. In fact, when they've done experimental infections early on, cats will show a leukopenia as maybe their only clinical response to infection. And most of those animals do find resolve and never show clinical signs. One of the areas that's interesting is that they do develop antibody responses to infection and vaccination. And this is a graph of a study that we did a number of years ago in domestic cats. The y-axis is antibody titers, and the x-axis are weeks after vaccination. So in this situation, we vaccinated them with a modified live vaccine. I'll describe that a little bit better later. And so at the initial vaccination, nobody had titers. 4 weeks later, we had very mild titers coming up in the animals. The blue line represents animals that received the vaccines subcutaneously. The red line down here is our negative control. Our purple line here are animals where we administered the vaccine orally. And the green line is the animals that received it intranasally. What we're interested here is what we were interested here is to see, to start to develop if we could induce a humoral response with non-traditional administration techniques. So thinking about how we might want to give this vaccine to animals in the wild. And that's why we were exploring the use of intranasal. This was a regular commercially available injectable vaccine. We're exploring giving it intranasally and orally. But you can see that subcutaneously, these cats had a tremendous antibody response about 3 weeks after the booster vaccination. And in fact, even the intranasal cats were doing well when we had to discontinue, when we were done with the study at 9 weeks. I would not recommend giving cats intranasal vaccines. This is what one of our cats looked like. This is not one of our cats, but it sure is what they look like. The second time you come at these guys with an intranasal vaccine, they really remember it. So let's talk a little bit about canine distemper in wildlife and wild felids. So in 1994, there was an outbreak of distemper in the Serengeti National Park in Tanzania. It was well documented and very frightening because this is a large park, well visited, and it was the first time that this was recognized as a problem in African lions. A subsequent outbreak in the adjacent park, Ngorongoro Crater, occurred in 2001. In both of those, they had losses of almost a third of the animals. The animals showed up as this guy, emaciated. Animals would have seizures. Pathology included encephalitis and pneumonia. And genetic evaluation of the strains identified it as closely related to the local domestic dog strain. One of the things that's interesting in this with this outbreak is that several years after, they went back and looked at a variety of data, a colleague Linda Munson and crew. This graph is a little bit difficult to piece out. The top line is actually the ambient temperature. The y-axis here is temperature. The bottom line is actually antibody titers. And what they saw, or what they project, is that actually there were a number of silent, if you will, outbreaks based on serology, where titers had come up and a large number of animals were infected, but nobody died, and so it went, or very few animals died, and so they went unrecognized. In fact, the big outbreak, actually the top line, the big outbreak occurred here in '94. The big outbreak occurred in association with a couple of environmental factors, including a drought, buffalo die-offs, and heavy babesia infections. And that probably is what caused this high mortality event, at least in the Serengeti. So tiger range, you can hear the range for the animal seems to be for tigers comes very close to you folks. The Indo-Chinese animal is probably just a very short distance to your east. Tigers range throughout India. I put this map up because most of you may not be familiar with the remainder of the range. So now we consider the Indo-Chinese tiger to be a subspecies separate from the Malaysian tiger down here in peninsular Malaysia. The Sumatran tiger, which is an insular subspecies. Historically, there was the Javan and Balinese tigers that are now extinct. There was also a subspecies in South China around here that has become extinct in the wild, although there are a number of individuals in captivity. And then the area that I wanted to point out that you may not be familiar with is represented this box here, which is the Russian Far East. So here you have Japan, the Korean Peninsula, and the border between Russia and China. And in this area, the AMUR River defines the border between the two. And this subspecies, historically called the Siberian tiger, is now being referred to as the AMUR tiger. Well, in the Russian, as they call the Russian Far East, tiger deaths have occurred due to distemper since the early 2000s. I would say one thing on this map is a little bit disingenuous because while the animals may range into the whole area that's green, and on a closer examination, what you find is small pockets of populations that are actually isolated, and these are referred to as metapopulations. And that's the real concern when we talk about disease, because these metapopulations, which may not, maybe the entire population is 200 to 400 animals, but at any given metapopulation, it may be only 30 to 40 animals. And a population that small is going to be very susceptible to what's called stochastic events such as disease or severe climate, hurricane, forest fires. And it's those metapopulations that we really worry about from an extinction standpoint. Recently, that is to say in the last 4 years, there's been one leopard death. The AMUR leopard is a subspecies of the common leopard that exists and is probably the most endangered subspecies, possibly down to as many as 40 animals in the wild. Also died of canine distemper. There's a picture of the AMUR leopard. And again, the clinical signs seen by these animals were very similar to what's been previously described, is to say they have abnormal behavior, other neurologic signs, and lesions include encephalitis and meningitis. Interestingly, on these animals, in contrast to the Serengeti situation, there is no recognized comorbidity. So we don't, at least to date, we haven't seen that there's other issues involved in these populations. It just seems to be straight distemper infections. The strain from these animals belong to the Arctic clade, which is similar to the domestic dog strains that are circulated. But studies of the area, and my colleague Martin Gilbert has done extensive work here, shows that a wide variety of carnivores in that area are infected, including canids, raccoon dogs, which are canids, badgers, which are mustelids, as are sable, and domestic dogs. In fact, these may be so broadly, the number of potential reservoirs may be so broad that actually modeling suggests that vaccination of tigers is probably the only means for really controlling the disease in the tiger population. In India, there's been canine distemper reported in both lions and tigers. A serologic survey performed in 2007 found that almost 90% of the Asiatic lions in captivity were positive for antibodies to canine distemper. And that's those animals were not vaccinated, at least to the author's knowledge. There are popular reports of tiger deaths and anecdotal reports of deaths in captive tigers. And then in 2017, 2018, there was an outbreak of canine distemper in the Gir forest population of Asiatic lions, which was initially reported and then more recently well described in the Journal of Emerging Diseases. So in captive animals, we see that cases occur, but outbreaks are probably more common. We've seen these outbreaks worldwide. They've been in Europe, they've been in India, they've been in other areas of Asia, and certainly North America. The overall, I would say, neurologic signs seem to be either appear more quickly than they do in domestic dogs, or we miss the early subtler epithelial signs. And to date, the disease has been overwhelmingly in PANTHERA species. That is to say, the big cats, lions, tigers, snow leopards, leopards. A couple of cases in jaguars. Smaller cats, mountain lions, and Felis species have had very, very few reports. So talking about control of canine distemper, the first step is always going to be biosecurity. So separation of susceptible species from potential reservoir species. Vaccination of reservoir species is a possibility in some cases. But certainly in North America, for us, our major reservoir is the North American raccoon. And so keeping raccoons out of zoos and sanctuaries is our primary means of biosecurity. And that's difficult because like many of the animals that you have that would be considered pests or vermin in captivity, strict biosecurity is difficult to attain. Issues regarding vaccine, we talk about vaccinating reservoir species. Certainly vaccinating at the reservoir seems to be dogs, and the dogs are pets. Then going ahead and vaccinating dogs is a strategy. It's something we've discussed with regard to canine distemper in areas where red pandas occur, especially in the eastern part of Nepal. But the trouble is that if your reservoir species is wildlife, then you have difficulty actually getting coverage of your wildlife species. And we're going to talk a little bit about efficacy of vaccines, but probably single vaccination, that is to say one shot, is not going to be sufficient for most of these animals. So when we talk about control in captivity, we're usually talking about looking at vaccination. And I'm going to go through and talk about our vaccine types, killed vaccines, modified live virus vaccines, and recombinant vaccines. And especially the work that we've done with these vaccines in non-domestic felids. So when you come to vaccines, there's really kind of three big issues. Safety is a major issue. Efficacy, whether these vaccines are going to actually protect the animals, is an issue. And then actually, when I talked with and presented some of our vaccine data in India several years ago, it became apparent to me that one of the big questions that needed to be addressed, and was a big question by many people in the audience, is just who should be vaccinated? Because we'll go into the issues around that, each of these. So talking about vaccine safety, really when we're talking about vaccine safety, we're talking about whether the vaccine is going to have adverse effects. And one of the, side products of our current COVID pandemic that we're seeing is now we are a lot more aware about vaccinology, about the pros and cons of vaccines, and certainly probably not a day goes by without some kind of comment about safety and or efficacy of COVID vaccinations. In our situation, the primary, certainly the worst case scenario for vaccination is, or vaccine adverse effect, is vaccine-induced disease. And in fact, vaccine-induced canine distemper has been reported in several species. So the red painting, again, the poster child, but the very endangered North American black-footed ferret. It has been reported in African painted dogs, although not as widely. So all of to date, the vaccine-induced diseases have been caused by a modified live virus vaccine. And I'll explain a little bit more about that. I think most of you are aware of what those vaccines are, but essentially those are vaccines that have a whole virus in them. The good news is that to date, no vaccine-induced canine distemper has ever been reported in a felid. So we are in good shape with respect to cats. But there still is a general aversion in the zoo world, and I'd say in the general wildlife veterinary world, because of these experiences with red pandas and black-footed ferrets, against the use of a modified live vaccine in a novel species. So let's go back to the vaccine types. We're going to talk about each one with its pros and cons. So killed virus vaccines, in this situation, you have a whole virus which has been inactivated or killed in some manner. It is then usually mixed with an adjuvant and administered. The vaccine, the virus is unable to replicate within the animal. So the dose that you give the animal is the total amount of antigen that they're going to see. So these vaccines are considered dose dependent, volume dependent, if you will. Killed canine distemper vaccines have not been protective, and at least in the United States, and to my knowledge in the world, there are no killed canine distemper vaccines being produced. The major killed vaccine, virus vaccine that we see is rabies, which is quite immunogenic. But right now, there really are no killed canine distemper vaccines available. And so really killed vaccines with respect to most of our animals is not going to be an option. Killed canine vaccines are not an option. Now moving to the next vaccine type, which is modified live, or also called live attenuated vaccines. These are vaccines that contain living virus that have been modified or attenuated to reduce virulence, usually through serial passage through novel tissue cultures. So you have a distemper virus that was pathogenic or virulent to dogs, and then after multiple passages through, let's say, embryonated chicken eggs or other cell lines, that virulence has been attenuated. That vaccine has the advantage that because it contains a live virus, when the virus is injected, the virus replicates in the body, and that replication produces ample antigenic material for the animal to respond to. And as a result, you get a strong and actually long-lasting antibody response compared to other vaccines. One of our issues with our modified live virus vaccine is that most of these vaccines are polyvalent, which is to say they contain multiple agents. So the Vanguard vaccine in the picture here contains canine adenovirus, parainfluenza virus, and parvovirus. And in fact, monovalent vaccines are relatively rare. Monovalent canine distemper vaccines are relatively rare, in part because people that are vaccinating their dogs, domestic dogs, want to give them the broadest possible coverage of their major diseases. So I will say that as a kind of take home, and I'll repeat this later, a number of modified live virus vaccines have been used in non-domestic felids. I'm going to do a quick review of that now. So after the 1994 Serengeti outbreak, veterinarians in Kenya decided to test a vaccine on some captive lions. They used this Novavax vaccine, which I had mentioned before. It was a Merck product that was used the Understafort strain, a chicken embryo adapted strain in captive lions. And the good news is they didn't see any adverse effects in the animals that are vaccinated. They did see good seroconversion in the vaccinates, but they did not see seroconversion in the non-vaccinates, suggesting that the animals that were vaccinated did not shed virus that could be communicated to other animals. So that's a relatively small study. There are in the literature, and these kind of get hidden in abstracts, animals that were vaccinated with a monovalent vaccine in Germany, in a German zoo. These guys did fine. In Japan, a large number of animals are vaccinated with a modified live vaccine. That vaccine was produced in Japan, and I couldn't get any information out of it, but there were no adverse effects in these animals, and their titers were actually quite strong. And there have been 2 reports of the use of a modified live vaccine in carnivore species, a variety of species. In China, again, I couldn't get what the product in the vaccine was, but the good news is that in none of these studies, either in Kenya, Germany, Japan, or China, have any adverse effects been reported in felids. In North America, we actually, my colleague Tim Juroff and several others and I did a retrospective study of distemper vaccination in large cats. And looking at safety, we found that there were 106 large felids that had received a modified live virus vaccine, including 70 that were my cats there in East Tennessee. And the good news is out of those 100 animals, only 3 had adverse effects. 2 of those were very mild, as in lameness or soreness. And there was really only 1 adverse effect, and that was a severe effect, and that was in a snow leopard which received a multivalent vaccine and showed some vague neurologic signs that resolved after 5 days. So I think this is relatively positive. Snow leopards, I don't know if you're familiar, how familiar you are with them. They're another Indian species. If somebody's going to get sick, it's going to be a snow leopard or a cheetah. They seem to be much more delicate than any of our other Panthera species. When we did have 1 adverse effect when we were studying this disease. We had an opportunistic, which is to say unexpected breeding at a facility, and that gave us the opportunity to vaccinate a pregnant female. She was vaccinated with a modified live virus vaccine that contained distemper and also contained parvovirus. And she gave birth to 3 full-term cubs, but unfortunately all 3 cubs died within 3 months of birth of congenital heart defects. And we couldn't find any agents, but we presume, based on a variety of information, that these were due to the canine parvovirus component of the vaccine and not the distemper component. So this use of these vaccines in pregnant animals, any vaccine, non-approved vaccine in a pregnant animal, is precarious, and this one just shows how precarious it can be. We're talking about efficacy. We're really talking about, will this work? Will this protect the animal? And the gold standard in efficacy, if you're doing domestic dog studies, is challenge. So you vaccinate an animal, you have some unvaccinated animals, you challenge both with virulent virus. The animals that are unvaccinated succumb to disease, and the animals that are vaccinated are protected. This is something that we're not going to do in any of our zoological species or our endangered species. It really is not something we can do ethically or want to. Another, most cell-mediated immunity is actually very important in protection for canine distemper. And if you could assess cell-mediated immunity, it would be wonderful. But it is cell-mediated immunity is very difficult to assay. They're expensive. They require live cells, and unfortunately, this isn't something that's widely done, although it would be very interesting. We've tried a couple of times and not been successful in coming up with a good practical cell-mediated immunity assay in our lab. As a result, most of the time when we talk about vaccine efficacy, we're talking about 2 things. A, are the animals getting sick? And that would suggest kind of anecdotal, because we don't know if they're being exposed. And then B, we're looking at antibody titers or the humoral response to either vaccine or infection. So antibody titers, they're widely used, relatively easy to perform, inexpensive compared to looking at cell-mediated immunity. You have to be very careful. I don't think you can compare titers lab to lab. One, you need to know how they're determining their titers. For most of what's considered kind of our gold standard, we're looking at serum neutralization, also called serum virus neutralization assays. But in those situations, you need to know what strain is being used in the assay, who's got it set up. And for all those reasons, comparison of a titer between labs probably is not appropriate. Another problem that we run into with vaccine efficacy is what is protective in a novel species. And even within domestic dogs, there's some debate. So there are papers in the United States that say if you have any measurable titer, that animal is probably protected. We have chosen in one paper to look at greater than 1 to 24 as a titer to be protective. Others would say greater than 1 to 32. And historically, greater than 1 to 100 was kind of used as a benchmark. So what constitutes a protective titer is another of the challenges that we have, and are probably going to be have to be defined by retrospective studies. So let's take a look at one. What we did, one study is we took tigers and we gave the Novivac modified live distemper parvovirus vaccine. This is the one I mentioned, that we had adverse effects in a pregnant animal. This is prior to that pregnancy. We did this, we looked at titers. So these are individual animals on the left. Initially, one animal actually had Chancard down at the bottom, actually had a 1 to 64 titer prior to the initial vaccination. Excuse me. But you can see at 3 weeks, all our animals had what I would consider strong titers. All of them were greater than 1 to 100. The next time we were able to bleed these guys, next time we were able to get blood from these animals was 6 months. Most of them still retained titers, although they drifted down a little bit, and most responded to a booster vaccination. So we were encouraged by this, and certainly from a standpoint of safety, it looks good. From a standpoint of efficacy, this is probably the strongest data to suggest that a one shot may provide some protection. When we look at, so in summary, we haven't seen any vaccine-induced distemper in any felid. Modified live vaccines should be avoided in select individuals. Pregnant animals, snow leopards being kind of the two that we've identified. And these modified live vaccines do provide very good and strong titers, even after a single shot. So the last vaccine type that I'd like to discuss is recombinant vaccines. Right now, the only commercially available one that we have is made by Boehringer Ingelheim, formerly Mariel. And these are the ferret distemper vaccine, a monovalent vaccine. The trouble is that there aren't that many ferrets in North America as pets, and certainly not as many as dogs. And so this has not got a lot of commercial value. There is a series called Recombitech vaccines for dogs. They make a variety of those, and those are the ones that we have looked at in red pandas, mostly because the PureVax vaccine has been marginally available. So these vaccines use a canary pox vector. If you're familiar with some of the COVID vaccines, they're using other vectors. In this situation, the canary pox they knock out the virulence gene in the canary pox virus. In that hole, if you will, in the genome, they then insert canine distemper virus H and F antigens or protein, genetic material, so that the animal then, once infected with the canary pox, produces the H and F proteins, and those are the antigenic material that then the animal responds to. The pox virus enters the vaccinated animal's cells, but it doesn't spread from cell to cell, and so that is actually a limitation of this product because it is essentially dose dependent. It doesn't replicate widely throughout the body like a more traditional modified live virus vaccine. The good news is that the vaccine doesn't contain whole virus, that is to say whole distemper virus, and so it cannot cause vaccine-induced distemper. And so this makes these vaccines attractive from a safety standpoint. Tim and I, we did a retrospective in North American zoos. Basically, these recombinant vaccines have been used in over 800 large cats, and they've been vaccinated almost 3,000 times. Most of these animals have been vaccinated with that PureVax ferret distemper monovalent vaccine. And the good news, the really good news, is there were only 13 out of this 30, almost 3,000 vaccinations, only 13 adverse effects, and most of those were very minor and could have been actually caused by other things. So most of these cats were darted for either immobilization or for vaccination, or were immobilized. And so some of the signs, lameness, vomiting, may have been more related to the actual anesthesia or the darting itself than the actual vaccine. So no serious adverse effects. This table from that paper shows antibody titers. In this situation, Tim decided to use a greater than 1 to 24 as what he would call seropositive. And you can see that at any given time, the majority of these animals, with the exception of the snow leopard down here, and some of the Sumatran tigers, although the numbers are small, majority of these animals had, at some point in time, had strong or seropositive titers. Probably the better table, I think, is this one, where you can see after, as the animals that had received one vaccine, relatively few of them were seropositive. After 4 or more vaccines, 3/4 of the animals had strong or seropositive titers. So that would, that would suggest that these animals are most likely. We did some recombinant studies, initially using the C3, the multivalent vaccine, and we also used the PureVac vaccine. In that situation, we actually gave it subcutaneous and on tonsils. Our data wasn't that inspiring. We didn't see any titers after the initial vaccine, 3 weeks after the initial vaccine. But after the second booster, we did have a few seropositive animals. Again, we were looking at oral vaccination or administering on the tonsil to see to try to assess whether this could be given orally. I think the good news is that no adverse effects, but not dramatic humoral titers. A couple of comments here, and I'm going to wrap up. We've done similar studies in red pandas using the recombinant vaccines, and the data that we've received is very similar. That is to say, relatively no adverse effects, small, short, or low titers after a single vaccination, but strong titers after multiple vaccinations. And interestingly, the recombinant series have slightly greater titers than those on PureVac. In India, there is Dr. Desai in Mumbai, is the Boehringer Ingelheim representative. This is her email. I actually emailed her earlier this week. She's still acting as a point person for acquiring the PureVac distemper vaccine. She said that the method for acquiring this vaccine has been made slightly more stringent. She's had a couple of requests, but very few people have followed through on getting a permit. But it is something that is can be done and is available in India should you find it necessary. And then I think talking a little bit about species and risk. One of the problems that many species are at risk, but what we see, and when I've talked to zoo veterinarians, relatively few people have seen clinical cases. If they've seen clinical cases, then they're more likely to vaccinate. But if they've been a veterinarian at a zoo for 20 years and never seen a case, they're asking, why should I vaccinate these animals? The risk seems to be fairly low. One thing that I have seen is that when an outbreak occurs, we usually see multiple cases. So usually isn't a single animal that goes down. It can be up to, we had an outbreak in a sanctuary in North America, 28 animals succumbed or became infected. So that's one thing. Usually one of the things that ends up happening is we end up vaccinating in response to an outbreak. And the other one is, the other question to ask is, what is your biosecurity? I visited the zoo in Darjeeling, the Himalayan zoo, and they have excellent biosecurity. And I think the veterinarians there, where the administration was reluctant to embark on vaccination of their red pandas, their big red panda population. But that is a situation where their biosecurity was very strong. In areas where your biosecurity is not as strong, knowing what the disease prevalence is in your reservoir species and how common those reservoir species is going to be another thing that's going to make you determine your risk. And then I think the other component of who should be vaccinated is cost. Vaccines cost money. Going through the process of acquiring them is difficult. And then vaccines have a relatively short shelf life. So that actually just increases the cost. So I think all of these things are factors in deciding whether or not to embark on a vaccination program. And I would say my own conclusion is every institution, every situation needs to make their own assessment. I'm certainly not promoting wholesale vaccination of all susceptible species. I think that has to be done on a case-by-case, institution-by-institution basis. Lastly, I talked just a little bit about my own interest, and here's one where I hope I might encourage a few of you out there to think about how do we deliver vaccines to wild animals? Certainly, injectable products. This is a mountain lion that I was involved in a wild mountain lion project many years ago in Central California. Certainly, if you're immobilizing animals for management reasons, they can be vaccinated. Some animals that are habituated may be prone to darting, but while we have what would be nice is to have a one and done, that is to say a single shot and you're good for life. That right now doesn't exist, but a monovalent modified live vaccine is probably our best option if injectable vaccination is an option. Certainly oral vaccines for wildlife have been developed. Orally delivered rabies vaccine is used successfully to control rabies in raccoons and fox populations in North America and Europe. We actually are right in the middle of, here I am again in Tennessee. East Tennessee is right in the middle of the enhanced surveillance area for rabies, raccoon rabies. And the purple bars here on this map show where the aerial distribution of the vaccine occurs. Actually, this little gap here in East Tennessee is where our national park is. So this is a strategy that is has potential. All of these vaccines, orally delivered vaccines, are vectored vaccines. And they've used pox vectors. The vaccinovirus for the oral rabies vaccine for raccoons. There's a raccoon pox virus that is used for plague vaccine in prairie dogs. These all actually use tetracycline as a marker, so you can come back and determine who actually consumed the product. And certainly an oral distemper vaccine would be desirable, but we got to be concerned about non-target species, such as raccoon dogs, carnivores, domestic animals, even people. And the cost in development, production, distribution, and monitoring are really significant. So for very isolated or small populations, oral vaccination is probably not going to be an option. I think as for tigers, I'm still thinking about things like sustained release products. They've been used in domestic animals, but not widely. And these would have a double vaccine or pellet where you'd have a sustained release. Another idea would be to use a system for transmucosal delivery. That is to say, delivering a product on the animal, which is then groomed off and acquired either intranasally or mucosally. Hopefully we'll think about these in the future. So in conclusion, I'd say that many modified live virus canine distemper vaccines seem to be safe in large felids. They elicit strong antibody titers. I would avoid the modified live canine parvovirus vaccine in pregnant animals. And the canary pox vaccines are safe to use in large felids, but multiple vaccinations are going to be required to produce titers. All this work has been done with a whole host of people. This is Dr. John Sykes from the Bronx Zoo. When he was a resident with us working at Tiger Haven. And I want to thank all these folks. And if there's time, I'm happy to try to take questions.
Okay, thank you, sir, for the elaborate presentation. So from the participant side, I feel some doubts they have typed out here. So I would like to read one by one.
Sure.
First discussion point, what they are telling is there always used to be a debate whether this canine distemper is fully cured or not. So in your experience, how do you feel whether it is fully cured or whether the animal will have some kind of shortfall or something like that?
I'm sorry, I'm going to work on, I'm going to go out of shared screen so that I can increase my volume, or try to.
Dr. Rajesh?
Yes.
Yes.
Speak little louder voice.
Yeah, I'm afraid I'm going to have to. Let me try this.
Am I audible, sir, now?
Yeah, I'm trying to. Oh, there we go.
Yeah.
All right. Much better.
Yeah. Yeah. Actually, one of the participants is asking whether this canine distemper is fully cured or not. So what is your opinion on this?
Yeah, I don't think canine distemper, so animals can resolve infections. So certainly what we see with, if you look back at the titers that we've seen on wild cats and to a limited degree on, there's a number of animals that have been infected because they have antibody titers, but have never shown clinical signs and never do show clinical signs. So animals can resolve infections. Typically, those infections are not showing clinical signs, and so you don't recognize that they even occurred. A small number of those will have CNS signs, central nervous signs later. Once an animal becomes infected and shows clinical signs, I think the debate is, I don't think that animal will ever be cured. And so the question, if you're a clinician, is how to handle that animal. And in a clinical practice, if an animal comes in with fulminant canine distemper, we are able to euthanize animals on a humane basis in the United States, and certainly many of those animals would be euthanized in private practices. If euthanasia is not an option, then you attempt to treat the secondary infections, secondary bacterial infections, and hope you can nurse the animal on. But I think the long-term prognosis is poor.
Okay, thank you, sir. Now the second question is whether the same vaccine which is used for dogs can be used for lions and tigers.
Oh, yes. Now, in fact, one of the things early on we discussed and explored the idea of developing our own vaccines. And as we've kind of seen with again, with the COVID situation recently, developing a vaccine is enormously expensive and time-consuming, difficult. So what we did is we explored the use of commercially available vaccines that were commercially available in the United States that we thought had the best potential for safety. And all of those have been commercially available domestic dog vaccines.
Okay, thank you, sir. Now, another question is, suppose there is an outbreak within a captive population involved in conservation breeding. And what you would recommend, putting the potential carrier down, or you suggest survival and the offspring will have resistance like that?
That's a difficult one. The answer is, I think when other places have had outbreaks, the question is how infectious is any given individual, and I think in other words, are individuals infectious lion to lion, tiger to tiger, or are they all getting it from raccoons? And I don't know that we have a good answer on that. I think you have to assume that any animal that's showing clinical signs is infectious and is communicable, and that animal should be isolated. Whether I think if that animal is showing clinical signs, if it's a novel species like a cat, I think it's going to die. I think it will be. I don't think it will be. I don't think it will live to be bred or part of the breeding program. So I think that's not going to be a question. Again, whether or not culturally euthanasia is an option in North America, we would probably elect to euthanize that animal to remove it as quickly as possible as a possible source of infection.
Okay, thank you, sir. Another discussion point is experience of canine distemper in fully vaccinated pups, especially the pups. Even after vaccination, they are showing the symptoms related to canine distemper, like seizures, all those. So what's your opinion in this regard?
Yeah, I think we have seen, there's been one case now. So first of all, I've not had that experience. So I've not seen canine distemper in fully vaccinated animals. What I have seen though is I've heard of cases of canine distemper in animals that have received inappropriate vaccines or poor vaccines. And I think that that's probably the area that that would be my suspicion of the problem that this individual is asking about. So we've seen in a snow leopard, again, snow leopards are problem animals, special cases. A snow leopard received one canine distemper vaccine, one of the recombinant vaccines, only one, and that subsequently developed a year later, developed canine distemper and died. The distemper that it developed was type to a raccoon strain, not the vaccine strain. So I think in that case, that animal, that wasn't a vaccine break, but that animal was inappropriately vaccinated. It wasn't protected because it hadn't received multiple vaccines. And I would guess that in vaccinated animals, I'd be very surprised if an animal had multiple vaccines, appropriate vaccines, that you would see a vaccine break. I can't talk for every vaccine. I'm talking now about what we would see in North America. The other issue, and I think this is one that would be under consideration for this, is if the mother had very strong maternal antibodies that she passed to the pups, then you may find that if you vaccinate them earlier than 7 weeks, that the maternal antibodies essentially negate the vaccination. And so you may see that if vaccines are started early, even if they received 2 vaccines, if the mother had strong maternal antibodies, that first vaccination may not be effective. And so that animal would be essentially, again, inappropriately vaccinated.
Okay, thank you, sir. In your experience, how do you feel, the combined vaccine or the CD vaccine alone? Which one is having more effectiveness, multivalent or?
Yeah, the work that we've done, there doesn't seem, with respect to canine distemper, there doesn't seem to be a difference. The one thing that we found with the recombinant, so when we're talking about the recombinant vaccines, the PureVac vaccine, which is a monovalent vaccine for the ferrets, it contains a lot more antigen just because as you add different components to the vaccine, like parvovirus and adenovirus, there just isn't enough room in 1 ml to include as much antigen of each. So in the zoo business in North America, we would say we would use the ferret distemper vaccine because it seems to have the most canine distemper antigen, canary pox carrying antigen. With respect to certainly feline panleukopenia is a parvovirus, and there's some debate as to how it originated, but using a parvovirus in cats is it probably assists in protecting them against feline panleukopenia. The trouble is feline panleukopenia is a virus that we know, a feline parvovirus, is a virus we know that causes congenital defects in pregnant animals, and that's where we really have where we have problems with that.
Okay, thank you, sir. Another question is, some of the vaccines, they are giving remarkably less titer, very less titer. So would you suggest to change the vaccine with another company? And usually the pups are vaccinated after 1 month, they will be giving the booster, then annually they will be giving the vaccine. But suppose if a pup is showing very less titer, would you recommend to vaccinate again?
Yeah, I think
with the same vaccine or a different vaccine like that?
Yeah, the answer is I don't think anybody really knows that. I don't know that there's strong. I don't know of any data that would support that. I can tell you that from discussions with virologists, specifically at DeBovie and Cornell, who I consider to be kind of the expert in this area, boosting a vaccination with an alternate vaccine seems to show much greater titers. So I think that is a. I guess I would say it is an option. I think the issue is using titers as a decision as to when to revaccinate is something that is difficult. I don't know that we have that really well defined because even an animal with a very low titer, let's say it's been a couple of years, if you revaccinate it, it may show a very strong amnestic response, which suggests that the protection is probably still there.
Okay. Thank you, sir. I feel these are the questions, important questions that is typed out in this chat session. So now I request the participants, if anyone would like to interact with the speaker, may kindly unmute your mic and you can proceed, please. If anyone would like to interact with the speaker, you are most welcome. Please unmute your mic and you can speak. I feel no one is there. So now I request Professor P.C. Kalidas sir for the concluding remarks, please.
Am I audible?
Yes, sir, you are audible.
Yes. Hello, everyone. It is really great that this international webinars, it's turned out to be very successful. As you know, almost 243 participants have attended the webinars. And now I am here to present a vote of thanks to the people who made this event successful. I would like to take a minute and thank all of them. First of all, I would like to thank our webinar speaker, Professor Eramsay, for enlightening us with his knowledge. Today's webinar was full of knowledge and interesting things. It gave deep insight into the topic and also revealed some interesting facts. The point where Professor Eramsay told us about the spread of canine distemper to various species, as well as its diagnosis, treatment, and finally the prevention by different types of vaccines available was really informative. I am pretty sure the precious knowledge that Professor Eramsay gave us will definitely help our colleagues, students, and participants in their studies and future career. Once again, I would like to thank Professor Edward Eramsay for taking out time from his busy schedule and enlightening us with their knowledge. Thank you so much, sir. I would also like to thank our Dean Madam for giving permissions to organizing this webinar session under the banner of NAHEP ICR and inviting Professor Eramsay to conduct it. I want to thank Associate Nodal Officer Dr. Ali, Dr. J.B. Rajesh, the Member Secretary, and other coordinator of the webinars. Special thanks to Dr. C. Bhanlal Puya, Research Associate, who really worked hard to make this event successful. I would also like to thank all the teachers for encouraging students to participate and also guiding the participants too. Thank you to all the faculty, students, and other participants present here for paying your attention and learning. I would end my speech here. Thank you, all of you. Good night.
Thank you, sir.
Thank you, sir.
So this international webinar is for 2 days. So the next speaker will be giving his speech on 28th July. So that is coming Wednesday at 3 p.m. to 4:30 p.m. And his topic will be applying stem cell biology and genome editing to poultry farming. So that will be from Dr. Mike McGrew, Senior Lecturer, the Roslin Institute, University of Edinburgh. So I request all the participants to come back on Wednesday at 3 p.m. to attend this speech also. Thank you all. Thank you.
Professor Eramsay, sir, please. Or yes. Hello.
Yes.
It's not visible, Dr. Rajesh. Eramsay, sir, is not visible.
Eramsay sir is already here. He is here only.
It is not visible.
No, he is here.
Okay. Yeah. I see. Sir, thank you very much.
It's been a pleasure. Namaste.
Thank you, sir. Yeah. Thank you.
All the best.
Thank you, sir. Soon I will contact you.
Sounds good. Take care.
Most welcome.
Saya pun kita upload din gitu. Kan tahu sih kalau semua. Kok kita ni hit boy tu. Ya, nanti don, don semua.
Okay, Dr. Rajesh, I think now we can end.
Yeah. Yes. Enough, no?
Recording stop.